Hello friends, this is Sajid, and in this video session we will learn about the Hybridoma technology. So this Hybridoma technology is used for the production of large number of monoclonal antibodies against a particular antigen. And these monoclonal antibodies are used for the different purposes. For example, in the treatment of cancer, and also as anti-venom, for example anti-snake venom, anti-bee venom, anti-spider venom. Basically these monoclonal antibodies are produced by a white blood cell which is called as plasma cell. Whenever any antigen enters in the body, especially the extracellular antigen, the B cell or B lymphocyte get activated by it and it is converted into the plasma cell. And finally, this plasma cell secretes the antibodies against that particular antigen. Now we might think, if the antigen is administered into the body of animal and that animal is going to produce plasma cell against that particular antigen which produces monoclonal antibodies against it, we can isolate this plasma cell from the lab animal and they can culture into the artificial media. And in artificial media, this plasma cell will produce antibodies for us. It is correct. The plasma cells can be cultured into the artificial media, but the biggest problem is that the lifespan of plasma cells is very short. It is from few weeks to few months. And that's why we cannot produce antibodies for a long time from culturing them into the artificial media. In Hybridoma technology, this problem of short lifespan of plasma cell is overcome by fusing it by a cancerous cell which is called as a myeloma cell. Myeloma cell, due to it is a cancerous cell, it is immortal in nature. So we have fused one cell which has the ability for production of antibodies and other cell which is immortal in nature. The new cell which is produced due to the fusion of these two cells is called as the hybridoma cell. And therefore this hybrid cell gains the property of both cells. So this cell is going to produce antibodies as well as it is immortal in nature. Let us see the actual procedure of hybridoma technology. First of all, the antigen against which the antibodies are to be produced is injected intravenously to the lab animal. This lab animal is generally mice or any other mammal. Since the antigen is given intravenously, that is directly into the blood, the antigens are trapped into the spleen. Spleen is the secondary lymphoid organ. Inside the spleen, the antigen interacts with the B cell and activates them to convert into the plasma cell. Once the plasma cells are produced, they will secrete the large number of monoclonal antibodies against the antigen injected. After this plasma cell producing the monoclonal antibodies against our injected antigen, they are isolated from the spleen. After the purification of plasma cell, it is fused with the myeloma cell. The fusion procedure is carried out in the presence of polyethylene glycol, and sometimes electroporation can also be used. Since the fusion procedure is random, it is not like that the plasma cells will always fuse with the myeloma cell, and therefore at the end of fusion procedure 5 different types of cells are produced, and they are fused plasma cells, fused myeloma cells, unfused plasma, unfused myeloma cell, and the most important, hybridoma cell. Among these 5 cells, we are only interested in the hybridoma cell, and therefore the next step is the selection of hybridoma. Selection is carried out on the HAT media, HAT. Here the H is for hypoxanthine, A is for aminopterin, and T is for thymidine. These are the contents which are present in the hat media. See, whenever any cell divides in the artificial media, before dividing it must synthesize the new copy of the DNA, and for that it requires the synthesis of nucleotides. Nucleotides can be synthesized by two ways. Either the cell can go for the salvage pathway or it can go for de novo synthesis. In case of salvage pathway, cells uses the degraded part of old nucleotide to produce the new nucleotides. And in case of de novo pathway, completely new nucleotides are synthesized using small metabolites like different sugars, different amino acids which are present in the media. In case of hat media, cells cannot operate de novo pathway, and this is due to the presence of aminopterin in the media. This aminopterin, it blocks one key enzyme which is required to operate the de novo pathway, and this enzyme is dihydrofolate reductase. Once this enzyme is blocked, cells cannot operate the de novo pathway. Okay. Therefore, there is only one option left for the cells in the hat media, and that is the salvage pathway. Cells can operate salvage pathway, but for this also there is one condition, and this condition is that cell must have one enzyme which is called as HGPRT, that is hypoxanthine guanine phosphoribosyl transferase. And for operating the salvage pathway, which have this particular enzyme, they can use the hypoxanthine and thymidine as precursor. Plasma cells have this enzyme, but myeloma cells don't. I want to remind you that 5 different cells are produced after the fusion procedure of plasma and memory cells, and these are fused plasma, fused myeloma, hybridoma, unfused plasma and unfused myeloma cell. Among these cells, plasma cells have HGPRT, and so the fused plasma cells. Myeloma cells don't have HGPRT enzyme, and therefore fused myeloma will also lack it. What about hybridoma? In case of hybridoma, one cell is plasma, and therefore it will have HGPRT enzyme. Correct? Now, what will happen if all these 5 different kind of cells are transferred on the hat media? Cells which lack HGPRT enzyme will not grow on the hat media, and these cells are fused myeloma and unfused myeloma. The other 3 types of cells can grow on the hat media, but lifespan of plasma and the fused plasma is very short, and within few weeks they will automatically die. No need to separately kill them. What about hybridoma again? It is HGPRT positive and it is immortal in nature. It will keep growing on the hat media. It has no limit. And therefore at the end, hybridoma cells will survive on the hat media. In this way, the hybridoma is selected and used for the continuous antibody production.